Hello, hello. So I think all of you know me already, but I suppose not. Most of you don't know what I've been working until July. So, as Yasin said, this is my PhD topic, but let me explain why we decided to study this topic in the first place. So we know that sociality is a very widespread phenomenon in the animal kingdom. Many taxa and many groups show some sort of social behavior or social structure. So one of the obvious examples is eusocial insects, but there's many different social structures shown by animals. And this is because sociality provides many benefits to animals that can go from thermoregulation, for example, to improved foraging efficiency or defense against predators and so on and so forth. But one of the most interesting groups, probably for the study of social structures, is mammals. They have been widely studied from this point of view. For example, there's a lot of literature on rodents, more specifically mice and rats, as we all know, and rodents are also the biggest mammal order. But bats are also like a pretty huge group. They are the second mammalian order in terms of number of species. They account for almost 1/4 of the mammal species, and they are very interesting. Let me show you why. So they are widely distributed. They are present in all continents except Antarctica. And on top of that, they show a huge diversity in terms of biology and ecology. Oh, I didn't realize I could use this. So here you can see, for example, the different diets that they can have. There's, as we all know, frugivorous bats, nectarivorous bats, but the vast majority of bats are actually insectivorous. But they can also hunt on small vertebrates. So in this case, you can see, I don't know if it's a gecko or a lizard, but also small birds. And we all know the hematophagous bats, so like the vampire bats, but those are a huge minority, meaning only 3 species. But other than diets, they also show a huge diversity in terms of hunting strategy, roosting strategy. They can use, for example, tree leaves, houses, caves to seek shelter, and so on. So they are a very cool group to study. But other than being highly variable, they show some, like just a few characteristics that are shared by all or most of the species. The one that is shared by all species is powered flight. That is what kind of defines the group in a way. But other than that, there's another one that is sociality. So you can see here, okay, that the vast majority of bats is highly social. So this is a graph that shows mammal species. Of course, it's not representative of the number of species because you can see that bats are a very small group. But on the outer side of the pie, the red bars, those are social species. The yellow ones are species that live in pairs, and the blue ones are solitary species. And you can see that for bats, the vast majority of species is social. But that implies, there's something missing, but whatever. That implies that they need to be able to recognize and communicate with each other. And to do that, bats use the 5 senses, the same ones we use, basically. But one of the other characteristics that is shared by most bat species is the fact that they are nocturnal. So they're mostly active during the night or crepuscular, let's say. And that makes hearing and olfaction fundamental senses in the everyday life of bats. However, while there has been a lot of interest going on around hearing or like vocal communication, when it comes to olfaction, there's very few studies on these topics. And we can see here a graph of papers. It comes from the Web of Science, and these are papers published in the different years. The orange bars and line is the regards papers on hearing or vocal communication, and the yellow one is olfaction. And you can see that there's a huge peak, like a lot of interest in vocal communication in a way, but olfaction lags behind a lot. But we know that olfaction is very, very important in bat sociality or bat social interactions, and that has been shown for many species. One of the classical examples is the sack-winged bats. So these are bats that live in the Americas, in Central America, I think, and they are called sack-winged bats because they have some sacs on the wings, especially males, and they collect odors all over their body. They kind of blend their own perfume and then they use it to court the females. So they fly in front of the females and they kind of like spread their perfume to convince them to mate with them. But there's a lot of other examples. So other than sexual selection, odors have been shown to be important for sexual discrimination, so discriminating females from males, of course, but also pup recognition. So imagine these like huge colonies, for example, of the Mexican free-tailed bats, where there's like so many pups and females go back, moms go back, and they have to find their own baby to nurse. It's not that easy, and olfaction or like odors have been shown to be really important in that sense. But the role of odors in the modulation and maintenance of the social structure is very, very poorly investigated. And that's the reason for my title, and also the reason why we decided to look a bit more into that. So this being a PhD thesis or like PhD project, it's for now divided into 3 chapters. I will go through the 3 of them quickly. So the first 2 I will present together because they are incredibly similar, you will see. And they are both around the impact of odor in roost exploration and social behavior. And why did we choose to do that? So I said that bats need to seek shelter, in this case during the day, because they are nocturnal, right? But most bats don't use the same roost every day. They switch very often, every 1 or 2 days, for example. This is especially true for forest-dwelling bats, that are bats that use, for example, tree holes or leaves to roost. But in order to do that, they need to continuously acquire knowledge about potential roosts in the vicinities and also be able to estimate roost availability and suitability, right? And of course, they can do it in different ways. And one of the potential, or like some of the potential cues that they could use is related to odors. And there's many sources of odors, right? So for example, gland secretion. So some bats are known to kind of mark roosting spots to be able to find them back. But there's a very widespread and obvious source of roost odor, that is poo. And the reason is that they go back after spending the night foraging, right? Okay, I will make it go away. Spending the night foraging, and they defecate a lot, and the feces tend to accumulate, especially inside the tree holes, for example, and they can be a source of many different informations related to diet, colony identity, individual identity, and so on. So we were not, of course, the first ones having the idea of studying these topics. There have been many people before us, and Bridget Brown and Gerald Carter wrote a review on this just a few years ago in 2022, and they looked into all of the studies that tried to estimate whether guano and urine, or like odors from guano and urine, are used by bats to select their roosts, find and select their roosts. And guess what? There are not too many studies. They found 8. Some of them you can see are pretty old, and they are on a variety of different species, but they are mostly insectivorous species. And they tried to look for a pattern here, and they found none. So in some studies, there seems to be some indication that bats can use or do use odors to find roosts. In some others, it's very clear that they don't. Some show like weak evidence, and some are very unclear. So it's a very confusing picture in a way, like what's going on. There could be many reasons behind this. One of them is that some of the studies are really old, so they have like outdated methods, or they lack stats, very limited sample size, and so on. But there could be many other explanations. So what we decided to do was to compare two species, and this is the Bechstein's bats and the network's bats. And the reason why we chose these two species, so they are both European species, they have a relatively wide distribution in Europe, and they are very similar in terms of ecology from many aspects. So they're both tree dwelling, so they use tree holes to spend their time during the day. They change roost regularly, so usually every 1 or 2 days they move to a new tree hole. The females are highly social, so during summer they gather into maternity roosts to give birth and raise the juveniles. And they are highly philopatric, so females always go back to the colony where they were born. But each colony shows fission-fusion behavior, so they split into subgroups, go back together, and then split again in subgroups with different composition. So they're very interesting to study, but the two species also, of course, show some differences. When it comes to Bechstein's bats, they show a very, very strictly closed social structure. So that means that females never move from one colony to the other, not even to visit other colonies. They always stay in their own colony. We know that because we have 30 years of behavioral data on them, so we're sure they don't do that. Yes, and the colonies can be incredibly close, so less than 5 kilometers apart and perfectly connected, but they just don't. There is some indication that they could use odor in roost selection, and more specifically to avoid parasitism. And they have been shown to be aggressive in some cases towards individuals of other colonies, belonging to other colonies. When it comes to nectar's bats, they instead show some intergroup connectivity, so they sometimes visit nearby colonies. They are not as closed as the Bechstein's, but they are still highly philopatric, but they can sometimes move and visit. And they have been shown to take roosting decisions based on heterospecifics. More specifically, they love, for whatever reason, to use roosts that have been recently occupied by Plecotus auritus, that is the long brown-eared bat. Yes, so what did we do? We have 2 study sites that are pretty cool. One of them for the Bechstein's bats is in the southern part of Germany, close to Würzburg. The other one is in the north for network's bats. And in these 2 sites, we have colonies where all females are pig tagged. So you know these tags that also cats and dogs have for individual identification? All females of our colony get one of those. And this means that we can monitor their behavior without really interacting with them. So we set out automatic loggers and boxes, and we are able to get a lot of data on the visitation of boxes, roosting behavior, and so on without really interacting with them. So what we did in both sites was to put up pairs of bat boxes on trees, brand new pairs and brand new boxes. And for each of the pairs, one box got feces of the same colony, and the other box got feces from another colony that was in the close vicinity, but with which the focal colony didn't have any contact. And what we did was just check what the bats did. You know, we just let the boxes up and saw which bats, when, and how they visited them. And we scored a bunch of behaviors, mainly linked to roost exploration and social behavior, but in this case, only for the nectar's bats. So what did we find? For the Bechstein's bats, we saw that they prefer to explore boxes that contain feces from their own colony. So these are all behaviors linked to box exploration, and you can see that in all cases they tend to explore more the boxes that contain feces of their own colony. And if you think about their social structure, it does make sense, right? Like they have this very close social structure. They tend to be aggressive towards individuals of other colonies. So it makes sense that they would tend to avoid odors related to foreign individuals, let's say. We also saw that this was stronger for fresh feces. So as soon as we put the feces when they're still like really fresh, and the difference tend to disappear or reverse the older the feces get. We can talk about that if you want. There could be a bunch of reasons. When it came to nectar's bats, we saw the complete opposite. So they tend to explore much more boxes with feces of a foreign colony compared to those with feces of their own colony. And I was quite surprised about this like huge difference, but again, it sort of makes sense if you consider that they move around and they tend, like they can visit colonies with like other colonies that are nearby. We also saw that when it came to social behavior, so they did explore more boxes with foreign feces, but when they did it, they tended to do it in groups or like in bigger groups and socially. Why that is, if that's because foreign individuals could attack them, or if it's just because boxes with foreign odors are more interesting, I don't know, but they tend to be much more social when exploring boxes with unknown odors, let's say. So wrapping up these first two chapters, feces odor seem to play a very relevant role in box exploration and social behavior. However, the direction of this like preference, let's say, or impact changes a lot depending on the species. And the reason why this is could be linked potentially to the social structure of the species that can be very different. And in this sense, roost odor could be in a way involved into the maintenance of this social structure, because if, for example, backsteinsbats tend to avoid boxes that have been occupied by other colonies, this could kind of like reinforce this closeness, right? And the other way around for naturalist bats. So the study on the backsteinsbats have been published. There's much more to it than this. If you want to read it, it's on animal behavior. And now I will move on to chapter 3, that is on a different source of odors, that is gland secretions, and more specifically the secretion of the interaural gland. So, as I said, odors and social behavior in bats have been not studied much, and this also reflects into the study of different glands and their role and their secretions and so on. So there's not much out there. But there is one gland that have been suggested to potentially be relevant for social behavior and more specifically individual recognition. That is this interaural gland. It's a gland that is placed between the eye and the nose here. So I don't know if you can see, but there's sort of like an orange dirty patch there, here, nasty one. And that's the secretion. The secretion is orange. That is pretty interesting. And the reason why this has been suggested to potentially be linked to individual recognition is that since a long time, there have been some behaviors that have been observed. And I will show you now a video. So these are two bats. This is a box, okay? And this is the entrance of the box that is on the bottom, and there's two individuals that are roosting already in the box. And now you will see there will be a third one coming in. So this is a very old video, it's from 1996. And you will see that the new individual sort of like greets the other two. And you will see what it does. So this is coming in, and see? So this has been described as like nose rubbing behavior, and it like goes on, and then it's like, oh no, that wasn't enough, and then goes back, and they greet each other again. So, and this is the reason why it has been suggested that potentially this gland could be involved into this behaviors and individual recognition. So, my supervisor, Gerald, and one of the first PhD students decided to look into this, and they published a paper on this gland. And what they saw is that the secretions of these glands are significantly different between colonies. So you can easily distinguish between colonies based on the composition of these secretions. And on top of that, they also seem to be highly individual repeatable. So the different individuals seem to have very clearly different secretions, and these seem to be highly repeatable over time. However, this study was done in a single year and during a single season. That is after the juveniles become volant, that is like late summer, basically. So in a span of a few weeks. So we decided to go back and look a bit more into it and try to sample across different years and run some other analysis. So we went back to the same colonies that were studied in that period, that are the ones in southern Germany that I showed you before. So, oops, okay, there are stuff missing, whatever, doesn't matter. These are the 3 colonies, and you cannot really see it from here, but they are pretty close by. So this is like 5 kilometers, this is like around 10 or 8. So they can definitely reach each other, right? But as I said before, they don't. And as I mentioned, we know a lot about these bats. So all of the females of these colonies have pictex, and all of the females of these colonies, and also the juvenile males that we can catch, but those leave the colony, are genotyped. So we take a wing punch from every individual, and we have the genotype, like we run microsatellites on them. So we know a lot. So what we did was basically repeat what had been done more than 20 years before. So we took a swab, and we swapped the gland secretion of the bats, and then we analyzed this secretion using gas chromatography and mass spectrometry, and from that we got the composition of the secretion. So like we got peaks, and each peak is a chemical compound, right? And then we analyzed this data. And we looked at first at patterns of variation. So we checked for differences between seasons and years, differences between colonies to try to replicate the results of the previous study, and individual repeatability, both long-term and short-term. And we did this using 3 different methods. The first one is PCA, and then we selected the 25 principal components and ran MANOVAs on them, and that's the same methodology that was used more than 20 years ago. Then we use permanovas that are more like a modern and widely used method to analyze this kind of data. And we also tried random forests, because for random forests, you don't need a dimensionality reduction. So you can use each single compound, right, and put that into the analysis. So it's a pretty cool method. And spoiler alert, but all of the methods agree on the results. Like there's no difference. They all show the same. So I won't show the separate results because of that. So when it comes to differences between seasons and years, we found both. So here, this is like a non-metric multidimensional scaling. So this means every point is a sample, and points that are more close by are more similar to each other than those compared to those that are more distant. And you can see that there's a pretty clear distinction between the samples collected in the 2 years. This is 2022, this is 2021. And when we look inside each year, but also if you analyze all the data together, we can also see pretty clear differences between seasons. So this is late summer, autumn, and this is spring. So we show very high temporal variation. Then when we checked for colony differences, we did find them. It's not that easy to see from this graph, but here, like this is a single year and a single season, because otherwise it would be way too much. So here is one colony, in the middle there's the other colony, and here's the third. And those differences are statistically significant in every case. So we confirmed what was found 20 years ago. Colonies are different in terms of gland secretions. And then we checked, as I said, for repeatability over time. We first checked on the long-term repeatability using the data from 2021 and 2022. And this is because we managed to sample quite a high number of individuals 4 times. So once for each season of each year, right? So spring 2021, autumn 2021, and so on. And we found nothing. We turned around the data in many different ways. There was no repeatability whatsoever. So we went back to the field in 2023 and we sampled the same individuals at 4 days of distance, because we were like, okay, in the previous paper they did find this high repeatability, right? So let's go back and try to see if it's a method issue or if the repeatability is there, but just on a short time frame, right? So we sampled those, as I said, those individuals 4 days apart, and we did find individual repeatability. So this kind of confirms the results of the previous paper, but we show that on the long term, this kind of disappears. Okay, and other than that, we also tested for potential links with genetic relatedness and behavior. So to do that, we took the gland secretions and we calculated the samples, and we calculated how dissimilar, let's say, they are from each other. So for each pair of samples, we calculated how similar or dissimilar they are. Then we also calculated genetic relatedness for each pair of individuals and roosting association. So we have all of the data from the PTEX. We just calculated how often they roost, every pair of individuals roosts together during the season, right? Or during the periods around sampling, let's say. And then we compared these 3 data sets, and what we found for genetic relatedness. So here you can see a graph, and on the y-axis is the similarity between secretions. So here on the bottom, there are secretions that are, or like samples that are very similar to each other, and these samples that are very different. And on the x-axis is relatedness. So this is closely related individuals, and this is distantly related individuals. And there is nothing, nothing at all. But that's actually good, because in the previous paper more than 20 years ago, they tried to do the same, and they found the same. So that confirms what was already found, that I guess is always good in science. Then we did the same for behavior. And here, this time on the x-axis, you find individuals that roost most of the times together, or individuals that roost apart, right? And what we see is a positive association. So individuals that roost together also tend to have more similar gland secretions, right? So wrapping up, we show very high variability in terms of gland secretions, both at the level of the colonies. So colonies are very different from each other, but most importantly, temporal variation. And we show no long-term repeatability in gland secretions at the individual level. Also, we show no connection with relatedness. That makes sense if we consider the temporal variation, right, and the lack of repeatability, and a positive association with roosting behavior. Whether this, so the association with roosting behavior, is cause of or consequence or both of this similarity is unclear. So like whether they tend to roost together more often because they smell similar, or the other way around. So the fact that they roost together more often makes them smell more similar, or if it's both at the same time, we don't really know, but it would be really cool to look into that. I would love to do that. So yeah, wrapping everything, like the 3 chapters up, we show that odours can play a really, really important role in bad social behavior, and this can be seen at different levels. So it can influence, for example, roost choice, it can drive roosting associations, and can help maintain the complex social structures that sometimes bats display. But I have an extra bonus chapter for you, because there's a lot of PhD students, a lot of them are starting, and I think in almost every PhD there's at least one part that doesn't work out, and you have to change the plans completely, and that applies to my PhD as well. And most of the times when I present the gland secretion experiment, people ask, but did you try to test it directly? You know, did you try to provide gland secretions to individuals and see if they react differently to them? And the answer is yes, we did think about it, and we did try to do it. So what we did, or what we tried to do, well, actually we did it, was to run choice test with a Y maze, right? So we had a typical Y maze at one of the, and like at the end of one of the arms, there was a swab with gland secretion from an individual of the same colony, and on the other, a swab with the secretion of an individual of a different colony. And then the bat was placed at the starting arm and could choose whether to enter the maze and how much to explore each arm. So very simple. We did do that. So this is a video. This is actually the best test we had. So you can see that the bat goes in and then kind of like checks around, and then spends some time, and then goes to the left, goes to the end, and you will see that checks around and then turns back and goes to the other arm. We ran 68 tests that, if you consider these are wild animals, it's not too bad in my opinion, most of them in summer. And bats emerged from the starting arm. That means like data that we could actually use in 45 of these, most of them in summer again. So I analyzed the data, and I found nothing. So there was no difference whatsoever. I quantified the different behaviors and there was no difference. So the first arm that was chosen by the bat, how much time it was spent, how many times it went back and forth, nothing, no difference whatsoever. And one could say, okay, like this is also a result, right? Like why would this be bad? Yeah, but because we know so little about these secretions and in general of odors, we actually don't know like how much odor is enough, how much the individual was stressed. Like, so did they, would they have shown preference if they weren't stressed, for example? Or like, do they need direct contact with the other individual to be able to perceive the odor because they couldn't really touch the swab in this case? So we don't exactly know what's going on. The original plan was to also have a blank, but for a bunch of reasons it didn't work. I can tell you why if you want. So we tried, we kind of failed. I think it's still relevant and interesting, so I would like to publish it even just to show people that this doesn't completely work, you know. But yes, but the most fun part of all of this is that we had a request from the German TV, the public German TV, to shoot some of these tests and some of this fieldwork for a documentary series for kids that is called Anna und die wilden Tiere. That means Anna and the Wild Animals. You can find the whole video there if you want. So the only part of my PhD that completely didn't work out basically is the one that was shown on national German TV that I find particularly funny. So here you can see a few snippets of it. It was actually pretty cool, and the bat behaved really well, so wasn't really stressed, at least apparently. So that was quite an interesting experience, and also serendipity, right? Yes. So as they say for raising kids, I also have the feeling that for a PhD it takes a village, and this is my village. So these are the 4 people in my supervision committee, and Gerald was probably the most important one. He made all of this happen. And then Saskia is a postdoc and supported me for the stats. Martina processed all of the samples. These are I don't have pictures for Malena, but these are the 3 students that collected some of the data for the first 2 chapters. And Franzi was super, like such a great student, and we set up the whole system together. It was great. And this is the research group. If you are interested in bats, they work on many different topics. They do really cool science. So please look up Gerald's group if you're interested. Yes, so I wanted to thank them all, and also everyone else that was to some level involved, like emotional support or whatever. And I want to thank you and the bats, you for your attention, and the bats for collaboration. And here you can see the Bechstein's bats. And there's this one cleaning the wing.